Hi everyone, we're going to go through the mitosis and meiosis lesson, and let me just put it in present mode. Okay, so mitosis and meiosis are two types of cell division, and we're going to start here with our essential questions. So here's the essential questions. Number one, what are mitosis and meiosis? Number two, what occurs at different phases in cell division? Number three, how are mitosis and meiosis similar and different? And so on the left, it's asking, if you were quizzed today, which questions would you know the answers to already? You're going to click and drag the green check mark next to the questions that you feel comfortable with. And then number two says, which questions would you need to learn more about to answer confidently? And you would click and drag a question mark next to the questions that you're really not sure of. So you can pause the video and do that now. All right, here's some vocabulary. We're going to go through the words first, and then I'll explain the diagram on the right. So a cell is the smallest structural and functional unit of an organism. Our bodies are made of them. Cell division is how cells reproduce by splitting apart. That's how we get more cells. A somatic cell are all the cells in the body except for germ cells. So somatic cells are everything except for sperm and egg cells in the body. And mitosis is the way in which somatic cells divide. If we need more body cells, they use the process of mitosis. Germ cells are only found in the ovaries and testes. That's where we get sperm and ova from, or sperm and egg from. So they're in the reproductive organs. And the way that germ cells divide is through meiosis. On the right-hand side is a nice, neat table that kind of organizes all these ideas. So the type of cell, if we look at somatic cells, they divide through mitosis. If we look at the number of chromosomes in a somatic cell, there's 46 in humans. There's different numbers for different organisms. And where can you find them in the body? They're found everywhere. The germ cell divides through meiosis, and there's 23 chromosomes in the germ cells, like the sperm and egg. And reproductive system is where you find the germ cells. So you're going to answer the question at the bottom, what is a cell? You should pause the video now. So a cell is the smallest structural and functional unit of an organism. All right, here's some more vocabulary. If we look at the two pictures, we're looking at the same DNA, but it's just in a different form. One of them is called chromatin, where it's unwound DNA. It's still found in the nucleus. And the other one is chromosomes, and it's tightly packed DNA, and it's found only during cell division. You can actually see it with a microscope. And it's easier for the cell to move these chromosomes around if they're all nicely, tightly packaged instead of all loose and unwound. So what is the difference between chromatin and chromosomes? You should pause the video now. So chromatin is unwound and a chromosome is wound up DNA. Okay, so now we've got some more vocabulary, and you should definitely be making some flashcards at this point to keep all these words straight. In a normal situation, chromosomes exist as a single chromosome. You get a copy from mom and a copy from dad, and they hang out next to each other. And so there's like this little button in the middle called the centromere. And one copy of a chromosome, if you look at the picture here, is called a chromatid. Right before the cell needs to divide, it has to make a copy of every chromosome. And the chromosomes kind of are attached in the center, the copies are, by the centromere. And each side, now that there's two chromatids, we call them sister chromatids. So over here on the right, a chromatid is each of two thread-like strands into which a chromosome divides during mitosis. And then sister chromatids are two identical copies of a chromatid. And the centromere is a structure in a chromosome that holds the two chromatids together. So our question over here on the left, what does a centromere hold together? You should pause the video now and answer the question. So a centromere is going to hold the sister chromatids together. You can see that round button-shaped thing here. All right, spindle fibers. These are going to control the movement and separation of chromosomes during cell division. A centriole helps in the formation of spindle fibers, and the nuclear envelope is the same thing as a nuclear membrane. It's a membrane that separates the nucleus from the cytoplasm in eukaryotic cells. So let's take a look at our picture here. These little orange rectangles are called centrioles. Okay, and when the cell is about to divide, spindle fibers start coming out, and this kind of like fishing line, and the centrioles are the fishing poles. And eventually, those spindle fibers are going to connect to the chromosomes, actually to the centromere, that middle part. And during cell division, these fishing poles, the centrioles at the end, are going to reel over, they're going to reel in those chromosomes. They're going to pull the chromatids apart and pull them to the ends of the cell. And in order to do all that, the nuclear envelope has to go away. That nuclear membrane has to break down so that there's room to move all these chromosomes around. Okay, so here's a diagram showing you several different types of somatic cells. So these are any cell that's not a germ cell. They contain 46 chromosomes, and we know they can divide by mitosis. So we find them everywhere in our body. This is a drag and drop. You're going to take the words and match them to their definitions. So you should pause the video now and work on this. Okay, the type of cell division that creates germ cells is meiosis. The type of cell division you find in your toes, since your toes are somatic cells, we're going to look at mitosis for that one. The loose squiggly DNA in the nucleus of a cell, that is going to be called the chromatin because it's unwound. One strand of a duplicated DNA, remember that's the chromatid, and a twin strands of duplicated DNA in the chromosome. Those are sister chromatids. The dot in the center of a chromosome, that's going to be called the centromere. Oh, hang on. There we go. All right. And then all the cells that aren't in a body that aren't formed in the reproductive organ are called somatic cells. Tightly packed DNA is visible with a microscope, that's chromosomes. And then cells that are only formed in reproductive organs, those are the germ cells. Okay, so mitosis here. Mitosis in somatic cells results in two cells that are exactly the same. They're going to be identical to the parent cell. And those two new cells are diploid cells. That means they have two sets of chromosomes. Remember that prefix di means two. And mitosis involves one set of division stages. So it goes through this cycle, but it only goes through it once. And you end up with going from one cell to two cells, and they're identical at the end. So we have mitosis is going to create new cells for growth and repair. So if you break a bone or get a scratch, that's how things get repaired. And currently you are in the middle of adolescence and you're growing a lot, and it's through mitosis that you're able to grow taller. So after a cell goes through mitosis, how many cells are there? You're going to pause the video and answer the question. It'll help if you take a look at this picture at the bottom. So there are two cells. You can see cell division results in two daughter cells. And mitosis is also used for asexual reproduction. We learned about all these in the last topic. So budding is the first one, and it's showing a hydra. It's this long center part. That's the parent hydra. And then it's got little baby buds growing off of that, and they're going to be identical to this parent. Number two is showing vegetative reproduction. There's offshoots underground that you can't see from the parent plant, and they're going to grow out, and you can see a new plant growing here. Number three is showing that this single-celled organism will go through binary fission. And then number four is showing that this organism can use fragmentation. It can be segmented into different pieces, and those will grow into identical adults. All right, so mitosis has four basic phases: prophase, metaphase, anaphase, telophase, and then cytokinesis is right along with telophase, right at the end. It does occur in a strict sequential order called the cell cycle. And it continues over and over and over. And it produces diploid cells with the same genetic makeup as the parent cell. So you're gonna have to know these in order. So make up a little funny saying or some way to memorize that they go in this particular order. You can remember it as PMAT. That might help you. But you should know the order that they occur in. And when we start learning what's happening in each of these phases, we're going to focus on what's going on with the chromosomes. And that usually helps for you to remember like what's happening. So interphase is a phase in between cell divisions. When the cell is not dividing, it's in interphase, and that's actually when it's doing its job. So if it's a skin cell, it's doing skin cell job. If it's a liver cell, it's doing the liver cell job. And at this point, it's still called chromatin because it's loose and unwound so that the cell can use the DNA for the instructions that are there. The cell can grow bigger. Usually right after the first cell division, it'll have to grow bigger. And the centrioles are going to be right near the nucleus. They're just kind of hanging out there. They don't have to do anything yet until it gets ready for mitosis. But right before mitosis is going to begin, or meiosis for that matter, the chromosomes are going to replicate. They're going to make a copy, and you're going to end up seeing the little X's with the sister chromatids. So here's your question that you're going to answer. Where's the chromatin during interphase? We're asking, like, the stringy stuff here, what is it located in? What organelle? You can pause the video and answer. So the chromatin is located inside the nucleus. You can see the nucleus is right here, and all the squiggly stuff in here is the chromatin. Prophase is the first phase of mitosis. This is when the chromosomes condense and become visible through a microscope. The spindles begin to form, the nuclear membrane breaks down, and the centrioles begin moving toward the poles. So a lot is going on in this phase. So first, the chromosomes are condensing. It goes from chromatin to chromosomes, and then you can see these little X's. The spindles are going to start coming out of the centrioles, and at the same time, the centrioles are going to move to opposite ends of the cell. So here's a question that you're going to answer. The nuclear membrane breaks apart to allow the contents to be used in mitosis. What is stored in the nucleus? So what can you find inside the nucleus? Pause the video now. You can find DNA inside the nucleus. We know that. That's in the form of chromosomes. Okay, metaphase is the next phase of mitosis. The chromosomes are guided by the spindle fibers, so those little fishing lines are moving the chromosomes to the middle of the cell. And I remember metaphase because metaphase and middle both begin with M. So that helps me remember where the chromosomes are during metaphase. And then the centrioles at this point are at the opposite ends of the cell. They're calling it a centrosome. That's an organelle near the nucleus of a cell that contains the centrioles and where the spindle fibers are coming out. So they're kind of calling it, it's the same thing as where the centrioles are. In anaphase, this is the third part. The two sister chromatids of each chromosome are pulled apart by the spindle fibers. So anaphase and apart are both going to start with the letter A. It'll help you remember what's going on with the chromosomes. The chromatids move away from each other toward the poles, and now each one is called a chromosome. The cell elongates so that the poles are farther apart. So you can see at the top that they're not connected like sister chromatids anymore. They've been pulled apart, and they're being reeled in by the centrioles and pulled to the ends of the cell. You can see the cell is getting longer, more egg-shaped, because eventually if you draw a line down between these chromosomes, it's going to divide into two separate cells. Okay, so here's our question. Why does the cell get longer during anaphase? You can pause the video and answer. And the answer is that way the cell can more easily split into two cells, and both of the cells will have identical copies of DNA in them. Now we move on to telophase and cytokinesis. This is the last phase of mitosis. The chromosomes have reached the poles and begin to unwind. The two nuclear envelopes now are starting to form in each of where the separated chromosomes are. So you'll end up seeing two new nuclei forming. There's a cleavage furrow. That's just the indentation in the cytoplasm or in the cell membrane that's eventually going to completely separate, and you'll have two cells. When it completely separates, we call that cytokinesis. Cyto is referring to the cell itself, and kinesis is just like the movement of it. So it's referring to the fact that the cell is going to split into two separate daughter cells, and they are going to be identical to that parent cell. Okay, so on this picture, let me go out of present mode. You are going to click on this handle, and you're going to be spinning it so that you're matching up the picture with the phase, what's going on in the phase. So I'll let you pause the video and see if you can match up the picture with the phase. Okay, so here is how these images match to the terms. Interphase is the rest phase, and you can see the chromosomes are kind of unwound here. They really should be unwound a bit more so that you can't see them in the form of chromatin. But then it goes into prophase where they have duplicated. And then metaphase, you can see the chromosomes are lined up in the middle. In anaphase, they're being pulled apart. And then telophase and cytokinesis, we're starting to see two separate cells forming. All right, our next click and drag activity. You're going to read the descriptions and try and match the phase that it's describing. And then down here in the smaller circles, you're going to put what order that phase comes in. So the first one here, it says, in which phase do the chromosomes line up in the middle of the cell? You're going to figure out which one of these it's talking about and what order it occurs in. So you can pause the video and do that with all the phases and the numbers. Okay, so in which phase do the chromosomes line up in the middle of the cell? Remember, middle starts with M, and so does metaphase. In which phase does the cell divide into two daughter cells? When we have two separate cells, that's at the end, and that's the T part, telophase. In which phase do the chromosomes move toward the poles? When they start to move apart from each other, that's anaphase. And in which phase do the chromosomes condense? That's at the beginning, that's in prophase. Down here, they're giving you remember PMAT is a way to remember the order. So maybe that'll help with the numbering. So here's the order that you should have. The first phase is prophase, metaphase, then anaphase, and then telophase. Mitosis was named in 1882 by a German scientist. The name was taken from the ancient Greek word mitos, which means string. What parts of mitosis may have inspired this name? So go back through the slides and read, which ones we're talking about, string or thread or anything that looked like that. And maybe you'll find that it kind of makes sense that they used this term. So you can pause the video and go back and look. So I thought maybe the chromatin, when it's unwound, looks like string, or the spindle fibers kind of look like real thin threads, and that's maybe why they use the term mitosis. Okay, meiosis is going to occur in germ cells. It's found only in the reproductive organs. The spermatozoa, that's the science term for sperm cells, and ova, or ovum, that's singular, ova is plural. Those are egg cells, and they each have only 23 chromosomes. They divide through meiosis. So which cells do you think are more common in the human body, germ cells or somatic cells? So if there's two types of germ cells and there's hundreds of types of somatic cells, I'll kind of leave that hanging in the air and let you decide. You can pause the video and answer the question. So yeah, somatic cells, there's way more somatic cells than there are germ cells. All right, cell division by meiosis. This is kind of a general overview here. Meiosis in the germ cell will result in a variety of genetically different offspring, and it involves two sets of divisions. So you can see here we're going from one cell and it's an interphase here. It goes through and it copies itself. So then you have these copies. They're in sister chromatid pairs like that. They go through one cell division. You end up with 2 cells. And then they go through another cell division. You end up with 4 different cells. And we're going to call them haploid because they're going to have half the DNA. You can see these each only have 2 chromosomes when we started out with 4. Meiosis reduces the number of chromosomes in the parent cell by half. It produces 4 gamete cells, and meiosis is required to produce ova and sperm cells for sexual reproduction. So what is meiosis? I'll let you pause the video and put that into your own terms. So meiosis is a process of cell division that creates 4 gametes, and they're going to be haploid and all genetically different from each other. Okay, here's some more vocabulary. Homologous chromosomes. During meiosis, the matching paternal from the dad and maternal from the mom chromosomes pair up inside a cell. So what happens is in humans, we have 46 chromosomes. You got 23 from mom and 23 from dad. And each chromosome has hundreds and hundreds of genes on them. So for scientists, in order to keep track of all that, they numbered all of the chromosomes because they're relatively similar between each person. You can find the same gene on each particular type of chromosome. So they number them. And so you have a chromosome number 1 that you get from mom, and a chromosome number 1 you get from dad, and a chromosome 2 from mom, and a 2 from dad, and so on. So the chromosome 1s are similar, and they hang out together, and those are the homologous chromosomes. The prefix homo means the same, and logos means information. So homologous chromosomes have the same information or similar information on them. Crossing over is a process where these homologous chromosomes are going to pair up, and then they exchange genetic material to form new combinations in those new chromosomes. And they kind of swap DNA with each other. And then the term haploid is talking about the 4 cells that form as a result of meiosis. They have half the DNA or just one set of DNA, and they're going to be different from each other and from the parent cell. Okay, so here's our overview of both parts of meiosis, meiosis 1 and meiosis 2. It's explaining both sets of cell divisions here. We're going to break it down and go through it in a little bit more detail. We're going to start with meiosis 1. So with meiosis 1, it starts with prophase 1. Now remember, interphase has already occurred, and the DNA has been copied by this point. The chromosomes now become visible under a microscope because they're condensing. They're going from chromatin to chromosomes. The homologous chromosome pairs align and crossing over is going to occur. The nuclear envelope disappears and the spindle fibers are going to enter the nucleus. So you can see how the pairs of homologous chromosomes are next to each other and they're crossing over. They're swapping DNA right now. This does not happen in mitosis, only for meiosis. So what is the role of spindle fibers? And if you don't remember, you can go back and look at the definition from one of the earlier slides, but you should pause the video and answer this question. The role of the spindle fibers is to pull the chromosomes apart or the sister chromatids apart and move them around during cell division. All right, so here we're looking more detailed at crossing over. This is only going to occur in prophase 1. And you can see that the chromosomes are swapping genetic material. It creates more diversity and variety in organisms. So what they're showing here with the letters is that like this blue chromosome has capital genes. These letters represent the genes on the chromosome. So it's got capital ABC. And then the red one showing a different version of the gene, lowercase ABC. And then they go through crossing over where they swap chunks of DNA. And now these 2 new chromosomes are completely different than the first original ones. They have different combinations of genes. And that's why there's so much variety with sexual reproduction. Not only are you getting genes from 2 different parents, but they also swap it out a little bit and they mix it up and you end up with a completely unique combination. So now you're going to click and drag and move the names to indicate what the picture is showing. So you should pause the video and do this activity now. Okay, so this first picture is showing a pair of chromosomes here. So that's our homologous pair. And then you can see when they make this little X shape next to each other, that's showing crossing over. And then here at the end, we've got our four new, they're representing the gametes or what would end up in the gamete cells. Okay, metaphase one, we're moving on. We just went through prophase one, and that's where crossing over happens. Now, metaphase, again, the chromosomes are in the middle. However, they pair up in the middle. They don't line up single file in the middle. That's where it's different than mitosis. And the spindle fibers are now attached from the centrioles to the centromeres because they're moving the chromosomes around and they're eventually going to pull them apart from each other. You're going to pause the video and answer what crossing over is, just kind of a review for you. The crossing over is when parts of homologous chromosomes trade DNA. Okay, on this table, it says the names of the phases of cell division were inspired by ancient Greek words. For each of these prefixes, describe why that Greek meaning is a good fit for the phase that it names. So, for example, with prophase, that's the beginning part of mitosis. The prefix pro means before or in front of. So prophase is a good name because it's telling us that it's the first phase in front of all the other phases. And then we have metaphase, and the prefix meta means after or in the middle of. And you have to figure out why that is a good name. Why metaphase makes sense. And then anaphase and telophase. So please pause the video and write down what you think. Okay, so meta is a good explanation because it's in the middle. It means in the middle of something, and that's where the chromosomes are. They're lined up in the middle. Anaphase is a good explanation for this part of the phase or this part of the cycle is because the chromatids are returning to their own ends of the cell. So ana means again or return. Telophase, so telo means the end or completion, and it makes sense to use telophase here at the end of mitosis because that's the last part of cell division. Then we move on to anaphase. Anaphase 1. It's the third phase of meiosis 1. The cell starts to lengthen. The two of each chromosome pairs separate and are pulled apart by the spindle fibers toward the poles. In meiosis 1, the chromatids remain together. And this is in contrast to mitosis, where the sister chromatids are going to be pulled apart. So you can see the spindle fibers are pulling the homologous pairs away from each other. So the question here is asking to take a look, what remains together and what separates during anaphase 1? So you're trying to see when it comes to these chromosomes, what part of the chromosomes are staying together and what is being pulled apart. You should pause the video and answer now. So the homologous chromosomes are going to be separating, but the sister chromatids are still connected at that centromere. Then we move on to telophase 1. This is the last part of meiosis 1. You have, at the end of this, you're going to have 4 sets of chromosomes in these pictures here. And a cleavage furrow is going to begin to appear. And we have that the cell is ultimately, after cytokinesis, you're going to have the separation of the cytoplasm, and you'll end up with 2 separate cells at this point. So you're going to pause the video and answer, what is cytokinesis? So cytokinesis is the separation of the cytoplasm. Okay, now we move into a part called interkinesis. This is where the cell just kind of takes a rest. There's no DNA being replicated at this point. So we're going to just look and think, how many cells were there at the start of meiosis? And then how many cells are there now at interkinesis? You can pause the video and answer these questions. At the beginning of meiosis, we started out with 1 cell, and as you can see in the picture, we have 2 cells. So it's gone through 1 cell division. Okay, here we have another click and drag activity. So I'm going to let you pause the video. You're going to read the descriptions and then drag the name of the phase that it's describing to the top part, and then you're going to drag the numbers to the bottom so that you know what order they fall in. So the first one is describing telophase. It says, in which phase is the cytoplasm divided by cytokinesis? That's at the end in telophase. The next one asks, in which phase does the nuclear envelope begin to disappear? That would be at the beginning. That's prophase 1. In which phase do the chromosomes line up in the middle of the cell? There was our M for metaphase. And in which phase do the chromatids remain together instead of separate? And they're meaning when the chromosomes are moving away from each other, they're talking about anaphase. And then here's the order that they all occur in. PMAT, remember, prophase, metaphase, anaphase, telophase. Here's another click and drag activity. You're going to be comparing mitosis with meiosis 1. So you're going to read these little cards and move them to where you think it's describing or which phase it's describing. And one of them is going to be the same for both. All right, so our first one we're going to take a look at is sister chromatids line up during metaphase. The sister chromatids, because it's just the chromatids, connected with the centromere, that's where it's specifically talking about mitosis. And then we have daughter cells that are identical to the parent cell. That's going to be mitosis as well. Resulting in 2 daughter cells, well, that's what happens at the end of mitosis and the end of meiosis 1. Crossing over makes daughter cells unique. That only occurs in meiosis. And homologous chromosomes line up during metaphase. That's different than the sister chromatids lining up during metaphase because the chromosomes are paired next to each other with homologous chromosomes. Okay, now our second part of meiosis. Meiosis 2, we're going to go through these phases, and this is actually going to look pretty similar to mitosis. So prophase 2 begins with the 2 daughter cells now that we ended up with at meiosis 1. Chromosomes are condensed. The nuclear envelope is going to go away again, and the centromeres or centrioles have replicated and moved toward the opposite ends of the pole again. So it kind of has to do a little reset again, similar to prophase 1. Metaphase 2, this is where it looks similar to mitosis. The sister chromatids are now lined up in the middle. The centromeres are attached to the spindle fibers from the centrioles. And here's a question for you to answer. Are any sister chromatids identical? Why or why not? So I'll let you pause the video and answer this question. Take a good look at the picture. Okay, so to answer this question, you can see that none of these chromosomes are going to match with any others. They're all very different. And that's because of crossing over. They had swapped DNA way back in prophase 1, so now they all have different combinations of genes. Anaphase 2, this is when the sister chromatids are pulled apart by the spindle fibers. Chromosomes are going to move away from each other toward the poles, and the cell elongates so that the poles are farther apart from each other. Meiosis 2 with telophase 2, that's the last phase of meiosis. Chromosomes reach opposite poles. The cytokinesis is the separation of the cytoplasm is going to occur right at the end. And then you're going to have the new nuclear envelope or nuclear membrane come back, and then you have 4 haploid daughter cells that are all different from each other. So now I'm going to have you answer this question. Using these images, compare the chromosomes in the initial parent cell with the haploid daughter cells. You're going to tell me how many chromosomes there are, how they compare to each other. So go ahead and pause the video. Okay, so the way you could answer this is that you're starting with 6 chromosomes or chromatids in the beginning and end up with 3 at the end in each cell. And in each cell, they're all very different from each other. All right, another click and drag. So you're going to read the description and match the phase and then put them in the correct numerical order. So the first one, in which phase do the chromosomes line up in the middle of the cell? Middle starts with M, that's metaphase. And then in which phase do the chromosomes move toward the poles? That would be anaphase 2. In which phase has the cell divided into 4 haploid cells? This is way at the end, so we know that's telophase 2. And then in which phase do the chromosomes condense? That would be in prophase 2. Here's our numbers and how they should be ordered: prophase, metaphase, anaphase, telophase. Okay, quick summary. Meiosis is a form of cell division that results in half the number of chromosomes in gametes or sex cells, the sperm and ova. It maintains the same number of chromosomes from generation to generation, and it results in an assortment of genetic material passed on to offspring. The numbers on the right represent how many chromosomes are found in each human cell, in these germ cells during meiosis, and you're going to compare what happens to the number of chromosomes during meiosis. So you can see the normal initial cell starts out with 46 chromosomes, and it goes through interphase and replicates to 92. And then it goes through meiosis 1, and you end up with your first division. You back down to 46. But then it goes through meiosis 2 and divides again without replicating, and that's how we end up with haploid cells, how they have half the number of chromosomes. So we start with 46, it doubles to 92, and we end up with 23 after dividing twice. Okay, so meiosis is like an intricate dance of chromosomes. For each stage of meiosis, describe a dance move you could do to represent what is happening to the chromosomes and why it fits. So you can see the examples here. Prophase 1, they form a circle, slowly start to split up. Metaphase 1, they line up in fours or two pairs along the middle. Anaphase 1, the pairs separate, everyone moves to the two ends. So I'll let you pause the video and see if you can describe, sort of in dance move terms, what's happening in the rest of the phases. Okay, so here's what I just came up with. If yours are a little bit different, but still describe how the chromosomes are moving, then that works too. So telophase 1, spin and dance in my own circle. I have my own separate nucleus at this point. Prophase 2, forming another circle. Metaphase 2, they're lining up in single file in the middle again. Anaphase 2, the partners are going to separate to the end. And then telophase 2, spin and dance in their own circle again. So here's a comparison table of both mitosis and meiosis. So we know mitosis happens in body cells. It has one division. It creates diploid cells, 2 of them. And new cells are genetically the same to the parent, and there's no genetic diversity. In meiosis, this happens in sex organs, the testes and ovaries. There's 2 stages of divisions, meiosis 1 and meiosis 2. It creates 4 haploid cells, and they are not genetically the same. So this is where we get genetic diversity from. Both of them are a type of cell division. They both replicate DNA, and they both create new cells. So this is another click and drag activity where you're going to label, is it describing mitosis? Is it describing meiosis? Or can it occur in both? So you should pause the video now and try this on your own first. Okay, the first one says a final result is 4 haploid cells. When you have 4 cells, that's going to be meiosis. And the next one over, the cell division of somatic cells. Remember, somatic cells are body cells, and they divide through mitosis. During prophase, homologous chromosomes cross over. That only occurs in meiosis. Metaphase, the pairs of chromatids line up in the middle, and when they're paired, that's in mitosis. And if you think about it, in metaphase, in metaphase or meiosis 2, in metaphase 2, that's where they're lined up in the middle as well. Spindle fibers form. That's going to happen in both processes. In anaphase 1, sister chromatids stay together. Because they're putting anaphase 1, we know that this is going to have 2 cell divisions, and that's describing meiosis. The final result is 2 diploid cells identical to the parent. Well, we know that's mitosis. If it's resulting in cells having 46 chromosomes, that's the same as, all our body cells, so that's mitosis. Resulting cells have 23 chromosomes. They've already done that one for us. That's meiosis. The last step is cytokinesis. That occurs in both processes. It's preceded, which means something comes before. This DNA replication comes before. It does occur in meiosis and in mitosis. And cell division of germ cells, those are going to be divided through meiosis. Okay, some last questions here. In your own words, what are mitosis and meiosis? How are mitosis and meiosis similar and different? So I'll let you pause the video and answer those questions now. Okay, and question 3, what occurs at different phases in cell division? So explain what's happening at prophase, metaphase, anaphase, telophase. So you can pause the video and answer this now. And for this last one, you should go back to the beginning and take a look at the essential questions. Are there any questions that you still don't really understand? And you should write them down in this box for me. All right, and that's the end of our lesson.